Looks like we made it. It's one of the last sessions of today and things on the program. This only speaks to the fantastic program and the organization of this meeting, and I would definitely like to thank the organizers for putting up such a great conference this year and for giving me the opportunity to talk a little bit about what we are doing in our lab. And of course I have to thank you for sticking around for so long and listening to this talk. So as we are very well aware, some of the biggest biomedical challenges in our time come in the form of metabolic disorders, autoimmunity, cancer, neurodegeneration. We also know that very often they're considered site-specific, especially in the clinic, and not really as connected entities. But more and more data shows to the fact that long-term metabolic changes can lead to chronic inflammatory disorders. Chronic inflammation can lead to cancer. Of course, we also know consistent inflammation and metabolic changes lead to neurodegeneration and so forth. It's also not one cell type, one tissue that would be responsible, but it's actually a whole plethora of genes, environment, and aging, which all come together to orchestrate the origins of such a disease pathology. I mentioned it's not just one cell type, but it's in fact multiple organ systems, the immune system, metabolism in general, that are all connected in orchestrating the pathways that lead to pathology. This brings us to the question, and also highlights the main culprit of trying to find solutions to those diseases, is that it's very, very hard to dissect what is the cause and what is the effect of such pathologies. And if you think about the disease models that are at hand, and it's a point I'd like to make, is on one hand, we have extremely complicated systems like in vivo models. And yes, we can get rid of a cell type, we can knock out a gene. Very often, most of us who also worked with animal models or other models know that they're very quickly compensated for, which kind of masks the origins. On the other hand, cell cultures are rather simple and give a viewpoint into a particular cell type, but not its surrounding circumstance. It's not by chance that I left the middle side of the slide empty, where I actually think that's where multi-organ microphysiological systems could help us bring in some of those causal relationships and try to dissect and discern under very systematic and scalable ways the biological complexity of the models that we search for and investigate. A beautiful example of such a complex pathology is inflammatory bowel disease. We work on many different projects in the lab, but I chose this one specifically. The word for intestine is also gut, and gut in German means something good, and I found it fitting for a conference in Germany. So IBD actually is consistent, mostly discussed in the context of either Crohn's disease, which largely affects the small intestine, or ulcerative colitis, which largely affects the large intestine. But in reality, it's almost like 40 different sub-flavors of IBD that exist today. They affect both men, women. We know there's genetics at play, there's environment in play, there's the microbiota at play. And given the connectivity of the intestine to essentially any other important part in the body, it's not surprising that actually patients with IBD are significantly more prone to develop other diseases that are extra-intestinal, that have nothing to do with intestine. And here's just one of the many lists that we now know are connected to IBD. In particular, the liver, because the gut and the liver are best buddies. They like to hang out a lot. They promote each other's functions. So we do know that patients with ulcerative colitis specifically are more prone to develop certain autoimmune liver diseases or even liver inflammation in absence of autoimmunity. If you take a look a little bit at the mucosa of the large intestine in IBD or in healthy conditions, it's very important to kind of understand that the large intestine is composed of the epithelial layer, which houses its own immune cells, intraepithelial lymphocytes, and so forth. There's the lamina propria that house a more complex, diverse set of immune cells, and that kind of leads and connects to the portal circulation that leads to the liver. So what we know about IBD, we don't really know what triggers it or what starts it. It can happen even in children that are as young as 2 years old. We call it very early onset IBD. But hallmarks of it are definitely bear disruption, triggering of innate immune responses first, and then that brings in the big guns, adaptive immunity, both B cells and T cells, that orchestrate continuous inflammation. There's a couple of different hypotheses how the inflammation environment of the gut is connected to the liver. One of those would be molecular mimicry, where the hypothesis is that kind of T cells that are primed to react against certain bacteria, confuse bacterial antigen with our own antigens, and thereby attacking our cells. Dysbiosis are changes in microbiome that leads to bacterial dislocations, leaky gut. Another one is inflammatory changes in the epithelium itself and kind of inflammatory signals being drained to the liver. And of course, the last one would be lymphocyte migration, lymphocyte activation, and traveling between different sites. In my lab, we're particularly interested in the later two. So the question we're asking is, can actually multi-organ microphysiological systems be used to untangle some of those hypotheses? And I don't know, I have no idea, but it's the mission that we pursue to kind of explore if that's the case and if we can really look at microphysiological systems as models to study complex diseases. I've started this path unbeknownst 15 years ago when I first started to model gut immune interactions in vitro, and then later pursued studies with animal models, human models, and macaques to study how the local immune environment in the gut dictates systemic immunity. But where it really came together for me was when I joined Linda Griffith's lab as a postdoc, where we developed a model of the human gut liver-brain axis, including immune cells and tissue-resident immune cells, to study how microbial metabolites in the gut inform progression of IBD and Parkinson's, and I would invite you to take a look at some of the papers that came out of it. In that study and in those projects, what we did is we used the platform 3XGL, where you can find more details in the papers, but essentially it's a platform that connects a multitude of different transwells and liver chips. So the way we constructed the gut part of the model is we would grow colon organoids, which would be then disrupted into single cells, seeded as monoid layers. We would underlay them with dendritic cells and macrophages. You can actually see on the far left side on the bottom, there's a happy macrophage eating on some E. coli. That's how we know that they're actually functional. And then the liver part of the 3XGL is actually based on Professor Griffith's work from quite a long time ago, based on liver scaffolds, where an array of channels can be housed with primary liver hepatocytes, Kupffer cells, stellate cells, and that's how we composed it. I also mentioned that we added a number of immune cell populations to those interaction studies, and I would like to highlight a little bit more CD4 T regulatory cells and Th17 cells. Those are kind of a really beautiful system to kind of that can actually work as canary in a coal mine to tell you how much inflammation is going on. Why T regulatory cells produce TGF-beta that puts a brake on inflammation. Th17 cells are often known to kind of create to promote barrier function, but they also can quickly turn inflammatory. But kind of the take-home message from here is essentially that when we talk about CD4 T cells, those are not black and white populations, but rather they're all on a spectrum. Th17 cells can become Th1 cells. Th17 cells can become tolerance-promoting cells. So it's not so black and white as we often read in papers. So what would happen if we create a co-culture of epithelial cells that come from a patient that has ulcerative colitis and ongoing inflammation with diverse immune populations? So in that case, we had ulcerative colitis organoids or healthy organoids, which we then co-cultured only with dendritic cells and macrophages. So we would add the T regulatory and Th17 cells that I just mentioned. Even just by looking at the morphology of the epithelium from both donors, you can very quickly recognize the hyperchromatic nuclei in the ulcerative colitis donor and disarrayed barrier interactions. But long story short, once you add dendritic cells and macrophages to the epithelium, you will see a kind of initial kickstart of innate immunity, where essentially once you add T regulatory cells and Th17 cells, they will just take off as a rocket and quickly become Th1 polarized, highly cytotoxic T helper cells. So what happens if we take a ulcerative colitis gut model with its dendritic cells and macrophages, and we add that to a liver model and we connect them together? Actually, something very interesting happens. Rather than on my assumption that we would see liver inflammation, we see the opposite. We actually see that the liver says, they're there, and the gut kind of calms down a little bit. The reason why it does so is essentially has a lot to do with immune metabolism. The liver is one of the great sources for glutathione. Glutathione is one of the most important antioxidants that kind of puts a big brake on inflammation. And we know that in patients with IBD, actually you have less glutathione. It's also connected to rheumatoid arthritis and other conditions. So just to make the case that it doesn't necessarily mean that, you know, if you connect one inflamed model with another healthy one, that you will have just systemic inflammation. So what happens is we add T cells to this. Actually, what happens is that then we actually see the kind of phenotypes that we would associate with ulcerative colitis and IBD. We see a disruption of gut barrier, increased signaling correlated to IBD. Similarly, in the liver, we see albumin just flatlining, metabolism of liver function dies down. So it's only once we start to add T cells, we start to see this very systemic IBD profiles. There's a side note I want to make. We also did a large global metabolomic analysis of media under different conditions. There's almost no group of metabolites that's not affected by T cell function and vice versa. So I'd like to make the point that it's also a bit of a prime time to start thinking about how we compose media for this kind of interaction studies, and that metabolism should play a significantly larger role in these discussions. So in my lab right now, we tend to call ourselves the Laboratory for Human Biomimetics. What we do is we pursue a variety of different projects, but they all kind of, what they have in common is we collect various donor matched tissues from organ procurement networks. Once the tissues come to the lab, we store some of them as snapshots for benchmarking once we start to establish MPS models, so that we have something to revert back to. We isolate different immune subsets, we collect them separately, we convert epithelial cells into organoids, for example, or store primary hepatocytes for liver models and so forth. We then use a variety of different MPS platforms. I like to call us MPS agnostic. We work with devices that come from academic partners. We have industrial devices. We build our own devices if that's what's called for. And we construct the models based on, you know, different scientific questions, and use perturbation studies to kind of recreate the model that we're studying. I'm also starting to realize that we are rather running out of time. So here's a model perfusion vessel system where we essentially can connect gut and liver tissue with its own tissue-resident immune cells and perfuse it additionally with blood-derived T cells, all stemming from the same donor. We can then also pursue and actually activate the gut model in order to start to understand a little bit more about liver immunity. What we actually see that when we add all the immune components, that the gut barrier is still intact, but once we activate the barrier, we see a huge disruption, which leads also to a little bit to liver inflammatory conditions. I've selected a selection of cytokines that are important for this crosstalk, but I'll point your attention to IL-22, where it's a very important cytokine in IBD. That kind of has a dual function. It can serve to promote tissue repair, but it can also fuel additional inflammation. So we went back and did single-cell RNA sequencing on the diverse immune cell populations that are in the model, and you can quickly see that the immune populations within the epithelial layer, lamina propria or liver or blood, are completely separated. There's very little overlap, so they do have their own T cell populations. We can do T cell clonal analysis to see how they're connected between the tissues, and we've actually identified cells which we call mate cells to be some of those that are highly distributed across multiple tissues and share the same clonal sequence. So mate cells are a newly identified T cell subset. Their name is because associated invariant T cells. I will have to quickly skim through slides, but the point I wanted to make here is they're a very interesting immune cell population that does not recognize peptidic antigen, but it actually recognizes metabolites, which is kind of quite unique in the adaptive T cell world. We are now actually pursuing studies to develop models where we can study mate cell biology. Mate cell biology between humans and animals is very different. And actually, if we start to look in the models and look at what mate cells do in our model, we actually see if we look at gene expression and GWAS catalogs, they're highly correlated to both inflammatory bowel disease and autoimmune diseases of the liver. And we have specialized tools like MR1 5-PRU tetramers that allow us to isolate those cells and kind of challenge them functionally and kind of start to dissect what their role is in individual tissues. And we actually can show that in fact we can both activate them in a TCR specific way, but also in a bystander way, which means that essentially mate cells can be both TCR activated and bystander activated, and they can lead to a lot of inflammation. Right now we're doing assays to confirm whether MR1 restricted cells, like mate cells, can orchestrate cross-organ inflammation, and we're trying to determine the role of TCR non-specific T-cell activation in cross-organ inflammation. I wanted to have a cute slide about the future, but I think that's not necessary, as we all know what the future is. But I do have to thank all the people in the lab, the MIT troop with Linda Griffith that set me on this path, and all the founding agency. Thank you very much. There's maybe a time for a question. Happy to answer it.
Thank you so much, Martin. So anyone has a question?
Hello, thank you for a beautiful talk. I just wanted to ask if the single cell sequencing you showed us was from the cells recovered from the tissues or from the organ chips? Or
there's a weird echo on the stage.
Oh, okay.
What comes from the front speaker doesn't really come to the back.
Oh, I'm happy to. Everybody else heard my question. Again, okay. All right. I was just asking if the single cell sequencing that you showed was from the biomimetic devices or from the tissue itself.
So the sequencing is done on the donor match tissues, and which are then constructed devices.
So it's post-device. Do you know what is the overlap between the T-cell localizations and clonotypes compared to in vivo? So you found...
It's identical. It's straight from the patient.
I know, but they migrated to different layers on the platform, etc. So do you know if your, I guess...
How it's changing over time?
yeah, or how it changed from in vivo to your platform? Okay, great, thank you.
Thank you, Stefan, again.